Look inside a typical baby's room, and you'll usually find a crib overflowing with dangling, colorful, musical toys. Parents instinctively provide this constant stimulation because they assume it helps develop their child's growing intellect. Curious, it makes intuitive sense. But figuring out whether these everyday experiences actually alter the physical structure of the brain has puzzled philosophers and scientists for hundreds of years. In 1785, an Italian anatomist named Vincenzo Malacarne tried to find an answer. He took pairs of dogs from the same litter and birds from the same eggs and extensively trained one animal while leaving the other untouched. When he examined their brains later, the trained animals seemed to have more complex folds, but for reasons lost to history, his research was completely abandoned. By the late 19th century, researchers tried a different approach. They attempted to measure the outside circumference of human heads, guessing that a larger skull correlated with a lifetime of intense learning. Later research proved this was an entirely invalid way to measure brain development. For centuries, humanity carried this strong suspicion that our surroundings shape our minds. We simply lacked the technological tools to look close enough to prove it. That changed in the 1960s. The invention of high-magnification electron microscopes finally gave scientists the ability to assess the brain precisely, right down to the level of individual enzymes and neurotransmitters. Armed with this new technology, researchers Mark Rosenzweig, Edward Bennett, and Marian Diamond at the University of California, Berkeley set out to settle the debate. They designed a massive 10-year operation spanning 16 separate experiments. To eliminate genetic variations, they used sets of 3 male rats from the exact same litters, studying a total of 12 sets per experiment. By rigorously controlling the genetics, they could isolate the exact effect of the environment, finally shifting the idea of brain plasticity from a philosophical guess into observable biological data. The researchers randomly assigned the rat siblings into 3 very different living conditions. The first was a standard laboratory colony cage, where a group of rats lived together with adequate space, food, and social interaction. The second was the impoverished environment. Here, a single rat was isolated in a slightly smaller cage, placed in a separate room away from the colony, with absolutely nothing to do. The third condition was the enriched environment. 6 to 8 rats lived together in a massive space filled with a daily rotation of 25 different toys, essentially a rat Disneyland. The litters were kept in these distinct habitats for periods ranging from 4 to 10 weeks. These sharp contrasts in daily stimulation were engineered deliberately. If the brain was capable of physically growing in response to its surroundings, these extreme conditions would force it to leave a clear, measurable signature. To eliminate any subconscious bias, the autopsies were conducted randomly by code number, so the examiner had no idea which environment the rat had lived in. This chart shows the ratio of the cortex weight compared to the rest of the brain. The cerebral cortex, the outer layer responsible for memory, movement, and sensory input, became significantly heavier and thicker in the enriched rats. Looking closer at these results, the enriched group didn't actually grow higher numbers of brain cells. Instead, the individual neurons grew larger, and the synapses, the tiny gaps where electrical impulses pass from one cell to the next, expanded by 50%. They also found highly elevated levels of a specific chemical called acetylcholinesterase in the enriched brain tissue. This enzyme is crucial for the nervous system because it allows for much faster and more efficient transmission of impulses among brain cells. The rats raised in the toy-filled cage physically constructed a heavier, faster, and far more interconnected biological machine. When these findings were published, many scientists were highly skeptical. They argued that the brain differences were caused by entirely different variables in how the rats were treated. Critics pointed out that the enriched rats were handled twice a day while their toys were changed, whereas the impoverished rats were never touched. Others argued that the extreme isolation of the impoverished cage caused intense stress, and that stress, not a lack of toys, stunted their brain growth. The Berkeley team tested these alternative theories immediately. They handled a group of impoverished rats daily and left their littermates untouched, finding 0 differences in their brains. In other studies, they subjected standard colony rats to daily stressors like mild shocks or cage rotation. Again, they found no changes in brain development. To test the absolute limits of their theory, the researchers trapped wild deer mice and placed half in natural outdoor conditions and the other half in the enriched laboratory cages. After 4 weeks, the outdoor mice displayed even greater brain development than those in the artificial rat Disneyland. By systematically eliminating handling, stress, and artificial environments, the team confirmed their thesis. The complexity of interacting with a varied environment physically builds the brain. While scientists cannot ethically run these kinds of isolation experiments on people, modern medicine has uncovered parallel evidence during autopsies of human patients. When doctors examine the brain of a person who lived with blindness, they find that the portion of the cortex dedicated to vision is significantly thinner and less convoluted than someone with normal sight. Without the daily experience of visual input, the brain physically scales back that unused tissue. Marian Diamond later applied these principles to human aging. By studying individuals who remained intensely active well into their late 80s, she found that continuous mental exercise literally preserves neural tissue. Diamond observed that because nerve cells are designed for stimulation, the physical thickness of the cortex depends on whether a person remains curious and mentally active. In short, those who use their brains are less likely to lose them. This research provides a framework for social ecology, the study of how a child's entire social environment determines their biological development. Researchers Weiss and Bellinger found that the structural density of a child's brain acts as a defense against environmental pollutants. Because an enriched setting builds larger neurons and more robust neural pathways, it creates a biological buffer. Children in impoverished environments lack this physical reinforcement, leaving their brains more vulnerable to damage from neurotoxic chemicals. On the other end of the spectrum, the Berkeley research has been twisted by popular fads like the Mozart effect. This trend promotes the idea that you can rapidly boost a child's intelligence simply by having them passively listen to classical music. Passive consumer habits do not trigger neuroplasticity. True physical brain development requires the same rigorous, continuous, and complex interaction that the Berkeley team observed decades ago. Your brain is never a finished product. It is a highly responsive, living record of every environment you choose to explore.